I was going to present some of these basics and papers that have sort of shaped the field. But you guys have not read papers. That's the challenge. So I hope I have encouraged you enough to read some papers at least, right? That's mostly review articles. You guys need to learn to read real papers, okay? All right, we started this yesterday, so we're gonna do an hour or so. Let's say until 1:40, 1:45, okay? And then we'll go for lunch and come back at 2:30 or so. Okay, so this was a slide was shown yesterday. Chromatin accessibility is altered by ATP-dependent chromatin remodelers. I talked about this. Why is this important? Why is chromatin accessibility important? Absolutely. Thank you so much. I love it. At least in 5 days, I've taught you this, right? To be able to move the nucleosomes. If you don't move the nucleosomes, we cannot make the DNA accessible for anything for it to bind, right? The whole objective of this class in last few days has been to teach you how epigenetic control of gene transcription. I mean, obviously, there's a lot more to epigenetics, as I said earlier, and hopefully learn some of it. I actually hope one of the instructors will cover the non-coding RNA and the microRNAs and all, because that's an important component of epigenetics. I didn't cover that because I thought, other people will, but maybe something I can talk about tomorrow as well, at least in a little bit, 30 minutes or so, because I think if you don't get that, that would be a shame. So I think it's important for you guys to understand RNA silencing and RNAi, which is again an epigenetic mechanism. So RNA-based control of gene translation and RNAi-based control of gene transcription. So all at RNA level, very different. Because there's an RNA class that later on, I think one of the other investigators is teaching it. I hope they'll cover it, but I'm going to at least touch upon it tomorrow a little bit, just so you guys get the feel of it if, it doesn't cover it. Okay? So what most important thing here, again, for this class is, this hour is, that chromatin remodeling has to occur. So nucleosomes has to be moved. So all we're going to focus on is that, right? And what there are professional machineries in the cell that do this, which are called chromatin remodelers. Simple. So there are chromatin modifying complexes. Again, these are not my slides necessarily. These are slides that I pull up from here and there. Some of these I pulled up from the review article. Some of these are some other people's slides. So there are 4 major families of chromatin remodelers. Switch SNF family, the largest of them all. There was the first one to be discovered in Roger Kornberg's lab, I told you earlier, guy who won the Nobel Prize for RNA polymerase structure. But he also discovered this major thing, right? Chromatin remodeling machineries. Another one called ISWI family. They're both switch SNF. They were actually discovered in yeast. You guys know what is yeast? Okay. And the last one is INO80. Okay. My lab works on several of these. We work on INO80 quite a bit, but also other switch SNF. There are lots of people who work on these right now. We'll talk about that in the end. But bottom line is switch SNF. Switch SNF stood for a specific, just like Drosophila. In Drosophila, people discovered this by different phenotypes. In yeast, the phenotypes are, what is yeast? Do you guys know yeast? Unicellular organism, right? A unicellular, a single cell, eukaryotic system. Eukaryotic system, great. Prokaryotes are very different than eukaryotes. All I've taught you is eukaryotes. None of them is prokaryotes, right? There are prokaryotes have a lot of different protobiology. In fact, tomorrow I'd like to maybe teach you a little bit of plant epigenetics, which is very different again than humans. But even you guys do a lot of work on plants, it's probably important. Let me see if I can find things to put together something for tomorrow. So tomorrow's going to be a filler thing that I think are important maybe for you guys, okay? But basically, switch SNF was found in yeast. Yeast grow. Yeast produce, what do you know best yeast for? Fermentation, producing alcohol, whole alcohol industry, right? The one that makes the money. Remember Kingfisher, Vijay Mallya, and all of those people? He makes tons of money for lots of people. Well, it's a great thing. We all need relaxation, right? Alcohol's good. Anyways, sorry. Yeast. So yeast, the important thing is yeast phenotypes are observed by growing them in different media that have different kinds of metabolite problems. Okay, so you can try to grow yeast in uracil deficient media, and you can see what mutations may have occurred in the yeast that grew in there. Wild type yeast will require uracil and go fine, but if I made a media that does not have uracil and yeast grows in there, I can see what uracil, what genes can overcome that, right? And once I know that, then I can again screen for things that will make it dead again. You get it? So if what suppresses that gene, the same way at the Drosophila screens were done for different phenotypes, right? Did you get what I just said? You can grow them in specific media, you can identify genes that are vulnerable, and then you can screen for things that will make them not grow again. One way to do it, you can actually flip this over too, okay? And that's how some people have found things that are from epigenetic machinery. Switch SNP was found in a similar fashion. Okay, it's I forget, but this is one of the metabolic screens that was done for SWI SNP, and that essentially is some nutrient deprivation condition that they grow well in. Okay, the yeast, a lot of other yeast screens are done in temperature sensitive mutants. So you know yeast can grow only in certain temperature, and again you can find yeast that grows in temperature that are not normal, and then look for genes and then do screens to suppress that phenotype. Okay, the phenotypic screens, and that's why these are easy, right? Because yeast is easy to grow. Take some, grow them on. Tomorrow, 2 days later, you know whether the growth has occurred or not. You plate it on a plate. You can do it cheap, well done, right? Doing any screen in mice is absolutely devastating on your bank balance, right? Because it's very difficult. It's also not possible a lot of times. Now people have more done, those kind of screens using, thanks to CRISPR-Cas. Somebody is a CRISPR-Cas fan in your audience, right? So basically, but these were simple organisms. So these screens were done 3 decades ago, 4 decades ago. People still do them, but yeast is not as popular now. But because you've exhausted every damn thing you could. Yeast has 6,000 genes. So somebody did screen for all 6,000 genes and everything. They figured out the function of all 6,000 genes. Done. Great, right? Lots of learning for anything that is conserved in humans or otherwise. Switch SNP complexes are completely conserved all the way. And yeast, Drosophila, higher vertebrates, mammalian systems, all of that. Okay, cool. So most of these families are actually found in yeast and in us, in humans. And that's why it's important. So yeast has done great service to us, same way as the fruit fly, same way as the zebrafish, same way as our beautiful mice that you don't like to touch, right? I have killed about like 2,000-3,000 mice myself. It's fun, guys, come on. The life in your hand. I probably have killed like hundreds and thousands of zebrafish embryos. Somebody's watching. Not gonna be happy with me. What all you do for humanity, right? All the pop I have gathered. Anyways, so these are 4 families. This is how they look. They have interesting domains. All of these have this helic domain, DX domain, right? That's their catalytic domain. There's ATPase remodeling domain. Then there are interesting domains, bromodomains and HSA. They're ATP remodelers, so they will have to have a ATPase subunit, right? The enzyme that hydrolyzes ATP is called ATPase. So these are the complexes that are found in humans, okay? They have multi-protein complexes. I told you that, right? Chromatin modifying remodeling complexes are lots of them, multi-mega Dalton proteins, multiple of them together. You see how many subunit switches SNP has? On the left side, right? So let's talk about this. ATPase domains, BERG1, that's SMARCA4, okay? SMARCA2, SMARCA4. There are actin-like subunits on this. What is actin? Again, what is actin? Cytoskeletal protein, right? Why do you need a cytoskeletal-like protein in a chromatin remodeler? Really? Why? Why do I care about an actin-related protein in chromatin? Structure integrity of what? The complex? No? Maybe to use the cytoskeleton network or something like that in the nucleus to move these chromatin machineries around. Maybe. Maybe. That's my hypothesis. I know a lot of people worked on it. I don't exactly know what all they figured out so far, but that's there. You can check if you want. But that was very fascinating to me. I was in a lab which was chromatin remodeling lab. Okay? But basically, I think they figured that that has some role to do with nuclear architecture and all of that. So look it up. But if I find it, I'll let you know. There are other domains. There are other proteins with domains which are interesting, right? Chromo related. I showed you in the previous slide domains that contain bromodomains, proteins that contain bromodomains, right? We talked about this earlier too. You know bromodomains and chromodomains, right? What do bromodomains do? What do bromodomains do? Huh? I can't hear you, sorry. What do bromodomains do? Reads acetylation mark. Lovely. What does chromodomain do? Reads H3K9 methylation mark. What does a PHT domain do? Reads an H3K4 methylation mark. Okay? So what have we got in this? We got all kinds of things. Some are DNA binding domains, some are chromatin machine modifying reading domain, right? Modification reading domain. Some are ATPase domain. So they have got their role figured out. I'm going to read a histone mark. I'm going to bind to the DNA when I want to. And I'm going to dissociate ATP to find energy to move nucleosome, right? Boom. Great job. So nature is beautiful to build these things. On the right side, it says different kinds of switch SNP complexes. BAF, PBAF, neural progenitor BAF, neuron BAF, and embryonic stem cell BAF. How cool. What does that even mean? BAF is just another, BAF is a switch SNP complex. Okay, so don't worry about BAF. What is preceding it? There are some that are found in neurons. There are some that are found in neural progenitor cells. There are some that are found in embryonic stem cells. They're specific to that cell type. The composition of these is specific to that cell type. So the cells want to do chromatin remodeling activity in a cell type specific manner. They make a specific composition that will only be useful for that particular purposes. Remember I told you about bivalent domains? Yeah, they're found in, I told you that earlier. Stem cells. So stem cells might, BAF may want to have something that reads bivalent domains, right? So that it can actually correctly pattern you, correctly differentiate you. Cool? And neuron progenitor versus neuron BAF has similar role in neuronal differentiation. The guy called Jerry Capri in Stanford is very famous for this, produced some of the best things. He discovered SWitchNIF and my boss discovered RISC, which are all same family in 1998 or something. That was when this was discovered. This complex was discovered around 1997, 98. Okay, just so you guys get the reference of how far we are. They were discovered then, and we have recently found their roles in cancers. These protein complexes are mutated in multiple cancers. We'll talk about that a few. And they also are very important because when you have deficiencies in these, they make neurodevelopmental disorders. I told you that a lot of chromatin proteins are absolutely crucial for correct neuronal function. Okay? We'll hear a little bit more tomorrow. But I just find it fascinating. A lot of developmental neurodevelopmental disorders have this chromatin modifying and chromatin remodeling complexes messed up completely. Okay? And that's how diseases are found. So we can cure a lot of these kids by figuring out the functions of these. People are working on a lot of these, actually. All right, so I wanted to bring home the simple point by previous slide and this slide is, sorry? That is one. But what is the more important point? Sorry, thank you. The more important point I wanted to bring home to you guys is they contain bromodomains, chromodomains, and PhD domains, so that they can read histones, they can go on to do, they can read the histone mark. So this is, as I said in the previous slide, like previous classes, chromatin remodeling complexes are the end result, well, not the end, at least a key intermediate result in gene transcription, recruitment of them, so that we can move the nucleosome for Tata binding protein and RNA polymerase to come sit on the DNA and do the transcription, right? These people are needed. So these people have to read histone modifications. The histone modifications come before, if you want to sequely think about these things, right? Histone modifications will come before the nucleosome remodeling complexes. Nucleosome remodelers will read them, and then those will move the nucleosome, and then the Tata, etc., binding factor, etc., will come and do their job, and transcription will occur. Okay, cool. So sequence of events is what I wanted to get you guys to think. Is that's a simple concept I think I want you to take home in last 6 classes, if anything else, because that is fundamental to the whole thing. All these things are made in a way that step 1, step 2, step 3, step 4. Okay, so multiple levels of gene regulation, right? Gene transcription regulation. Cool? We know this, right? Okay, we'll read a little bit more, but we'll come to this again. SMARCA 2, SMARCA 2, 4, 2, 4. These are two things, SMARCA 4 and ARID 1A, 1B, ARID 2. These things are highly mutated in cancers, very highly mutated. SMARCA 4, this is a subject of lots of pharma world's investigation right now and drug development around SMARCA 4 mutants. Okay, and I told you the concept of synthetic lethality earlier, did I? Yeah, so SMARCA4 mutants are actually very sensitive to SMARCA2 deletion. So in SMARCA4 mutant cases, SMARCA4 targeting drug just cures them in preclinical models. Okay, meaning they're not yet FDA approved, they're not yet clinic, but they're all being tried in clinical trials. Okay, so same way with MTAP and PRMT5. I told you about MTAP deletion, PRMT5 at some point, right? Similar to the synthetic lethality. Okay, ARID 1A, 1B mutations happen a lot in ovarian cancers. SMARCA4 mutations happen a lot in lung cancers and multiple other cancers. PBRM1 mutations happen a lot in kidney cancers. I hope the cancer guy will tell you a lot more about it. People have developed drug against various parts, BRD9. People have built drug against SMARCA2, as I said earlier. BRD7. SMARCB1 is actually mutated in a lot of cancers. One of its kind is called rhabdoid tumors. SMARCB1 is the thing that is mutated. That's the only thing. So one gene that is deleted makes the cancer. Okay, like this ARID2 is mutated heavily in multiple cancers. So this switch SNF chromatin complexes are heavily mutated across various cancers. And then when mutated means they're dysfunctional. They don't function. Right? And if they don't function, chromatin remodeling does not happen, transcription does not happen well, and cancer becomes cancer. Okay? So in all of these classes, I've also told you that there's a lot of mutations happen in different parts of chromatin machinery that is regulating gene transcription. So this is absolutely crucial to the whole cancer world, right? Okay, so a lot of interest in the field, lots of drug development, billions and billions and billions of dollars really poured into these fields by many people. Okay, so two major types that people have looked a lot into switch SNF, CHD. I just belabored upon it, bromodomain, this chromodomain. There are two different kinds, right? They read acetylation mark, bromodomains. They're there in switch SNF, I mean, these complexes, and chromodomains CHD134, they read methylation mark. That's what I want to get you. I keep on saying the same thing, so it is in your memory. You know what I'm talking about, okay? So the CBAF or any BAF complex is recruits in H3K27 acetylation, which is your, the enhancer mark. I told you that earlier, did I? No. Okay, let me repeat that. Most of the enhancers are marked by something called H3K4 monomethylation first. Monomethylation marks an enhancer for poise. So it says, okay, here is an enhancer, right? And when it's combined with H3K27 acetylation together, then it marks an active enhancer, okay? Active enhancer is something that is go on to recruit everything else for the gene transcription to begin. And I also told you enhancers correlate the best with cell phenotypes. So in your eyes, in your skin, in your whatever cells, there's unique set of enhancers in every single cell type. Okay? That does not mean that, so, you know, eye has only 5,000 enhancer, and that is very different from skin enhancer. 5,000 enhancers would be there uniquely in eye, and 5,000 will be there in skin, but there'll be a set of like 20,000 more enhancers or more that will actually be common in both. Those are maintenance enhancers. But there is a set of enhancers that are unique and specific to each cell type. Okay, and you utilize that information to define what kind of cell type that could be. Okay, is that good? Okay, enhancers are absolutely crucial earlier for our understanding of development, right? And now many people have shown, including my own lab, that enhancers are absolutely crucial for cancer development, for define what cancer phenotypes are going to be. Okay. Okay, K27 acetylation is read by bromodomain that is found in the CBAF, and therefore this chromatin remodeler will move the nucleosome here, will allow these other things to come and bind right here, and that will make the active mark. This is the repressive side of things. There's some of these other BAFs, so CBAF is doing actively, so it's opening up for other binding for active transcription. Here it is opening, it is suppressing it. Well, sorry, here it's doing another job, which is reading this mark and now suppressing the actin of K27 as a trimethylation. So they have slightly different job. That's what the author is trying to tell you. CBAFs move nucleosomes, PBAF and CBAF move nucleosomes, plus, you know, suppress the repressive mark. Okay, that's what point was. Okay, so they have more than one function. They move nucleosome, but they also suppress the next kind. Okay, the repressive marks, they remove repressive mark to go on, and therefore they can start the gene transcription. I'm sorry. Okay, good. Is that clear? Okay. So they have, how do these things function? I think I touched upon this yesterday also. So they function by 4 different mechanisms. Sliding the nucleosome, cool. Move nucleosome, slide from here to there. Eject the nucleosome, so kick it out of the DNA. Ejecting specific parts of it, so they will remove H2, H2B, for example, okay? And break down the nucleosome, that will reduce the binding, you know, of the DNA on there, and then they'll replace it with a variant histone. And the variant histone then will be used for gene transcription or activation and recruitment of other things, because the variant histones make nucleosomes have different architecture. So they don't, in the H2A case, in this particular case, for example, H2A variant case, the nucleosome is not very tightly bound to the DNA anymore, okay? And it's not very stable itself. So it's much easier to get out, it's much easier to move the DNA. So that's one thing that they do. Is that clear? Did I make it myself clear, or you get confused about that? Just the variant composition, the composition of nucleosome itself will now allow the change in affinity of DNA to the histone proteins, okay? We'll talk about that a little bit more. So when the nucleosome sliding happens, you switch SNP binds, it hydrolyzes ATP, displaces the nucleosome, rebind it, okay? And forms a larger DNA bulge, DNA is rewrapped, and end, okay? Which means the sliding. So it essentially loosened it around it, the slide, and then again wrapped, right? Cool? That's sliding. Okay, let me see. I had a picture I wanted to show you guys. Where is that? Man, did I lose it? Oh, no. Because I want to show you guys an experiment. Damn, I made a mistake. Actually, it's a very important experiment, very simple experiment. Okay, wait. I'm gonna pull those slides up. Maybe I can just show you guys on my computer if I have them here. I think I deleted them by mistake. Oh, all right. Wait. I think in the process of creating this deck, I deleted like 3 important, very important slides. Hmm. Hmm. Okay, maybe I can show you guys this on my screen. You guys can see my thing. No, you can't see it, right? How do I open it here? Oh, I can open it. Yeah. Okay, let's see. Okay. Yes, mom. Chromatic remodeling first step is stone addiction. Yes. Then stone replacement and nucleosome sliding. Those are 3 different mechanisms. They're not first, second, third step. Some remodelers will do that. Some remodelers will do the other thing. Some remodelers will do all depending on the context where they are. Okay? Is that what your confusion is? No, not steps. Sliding is one mechanism. Yes. No, same thing. Ejection and eviction is same thing. At least that's my understanding. Are you confused about that? Think about them for your purposes to be same thing. There might be some technical differences. These scientists go crazy about their own names and all. So don't worry about it. Okay. You guys see an experiment on the screen, right? Okay. I'm not going to take you the whole thing. I want to show you two panels. First panel here. Can you see something called something DNA here? This doesn't project very well, unfortunately. You see, this says intact nucleosome. Okay. Don't worry about it. This cylinder is essentially a nucleosome. There is a site. So these guys, what they did was they put a DRA1 site here, a restriction enzyme site. Okay, and they're looking for cutting of the DRA1. Whenever DNA cuts, it will run here. When it doesn't cut, it will run here. So they cut it, then proteinase get treated, the nucleosome goes away, the DNA length remains. Okay, the DNA is labeled with radioactivity 32P. Okay, you guys know what is radioactivity? You guys know Mary Beckerle? Yeah, good. Mary Beckerle got 2 Nobel Prizes. See, I'm obsessed with Nobel Prizes. Okay, so very simple. They took a template, the DNA template, they positioned a nucleosome on it. This is all in vitro, meaning in a test tube. Okay, they isolated the risk nucleosome. Risk is a switch sniff yeast nucleosome essentially. Okay, one of the yeast switch sniff nucleosomes is called risk. This is what my boss had discovered. This is actually his paper. It's a paper of one of my seniors. His name is Anjana Basai. He's from IIT Delhi. And he's an amazing guy. I saw this work happening in front of me when I was a 21-year-old kid. And I was like, how amazing. This is beautiful science. That's why I'm showing it here. Okay. Anyways, this is nucleosome position. There is a DRA1 site. You can put the enzyme after the DNA is cut and DNA will cut or not cut. You can do this in a southern blot. Remember the southern blot? Okay, cool. So they say they're doing, what are they doing here? They're doing intact DNA. It's only DNA. When it says minus, means it's not there. When it's plus, it means there. The enzyme is there, minus, doesn't cut. Enzyme is there, cuts, nice. Like, cool. Don't focus on anything else. Just focus on this gel. Can you guys see this gel? Am I blocking your view? It's fine. You can see here. Oh, sorry. Of course, you can hear me. See. All right. So basically, as you increase the draw one, it'll cut. Now, risk is minus, minus, minus. It says 1, 4, or 2, 4, 8, 12, 16. They're increasing the amount of how much chromatin remodeler they're giving you. Okay. And then there's ATP, minus, 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 minus. If the ATP is there, no cutting. You know, that's good because it's ATP dependent activity, right? That's what ATP dependent chromatin remodeler means. ATP will work only when ATP is around because it requires ATP's energy to do what it's supposed to do. Okay, so now you see these lanes. It's nice and clean. There is very little or zero. Maybe this is zero actually. How bad is this? Oh no, that's 8. That's because there is no. This is okay. This is yeah. So 4. This is 8, and there is no ATP. So even this doesn't work. But if you say 4, you see there's a nice cutting. You can see a band in the bottom. So you're looking for the presence of the band in the bottom. Okay. Now cuts 4, 8, 12, 16. Complete cut. Right. Cool. Very simple experiment. What does it show you? What's your interpretation of the experiment? You can see better? Okay, yeah, we can see much better actually. I should have done that. So just focus on this and focus on this. What does the experiment tell you? Examination. Come on, it's easy peasy. This is a 6th grade question. Anyone? There are no wrong answers, guys. Say that again. Okay. Okay. Does everybody have any other interpretation? Yeah.
In the presence of RS and ATP, the cleavage is occurring, hence we can see the key in the bottom side. But in the only presence of RS, it is not occurring, so we can interpret that ATP is required to.
Good. Anybody else? What else do you see? Yeah. Okay, so that is actually is just a negative and a positive control. So that's just trying to show you where the band should run when you cut it. But that's a good observation. Thank you. What else? What else do you observe? So very simple experiment, guys. This shouldn't take this long. It's the simplest of them experimental. Anyone here on the right side? No? Okay, you want to go? Sorry, what's your name? The one behind you, the girl. Jyotika ji, do you want to tell me what you see in panel C? Just tell me what you see. Don't worry about it. It's okay. Sorry, say it again. Loudly. Huh? What? Of ATP molecule. Okay, there is a use of ATP molecule, but what do you see with the use of ATP molecule? What are the black things on the blot? Do you know this is a blot? We've talked about southern blotting. You guys said yes, sir, on everything. Come on. What do you see? It's okay. Tell me what you see. Okay, you want to come here? Let's help you out. It's okay. I want to make sure that you guys get it. Okay? Tell me what you see in panel C. So forget about everything else, right? Only panel A, panel C. Panel A is what do you see on panel A?
The intact nucleosomes and the nucleosomes are there.
Uh-huh.
Which one are the clear?
It's okay. It's okay. Don't stress out. It's fine. I know you guys coming on that stage is difficult for you. It's okay. Tell me, go there and tell me what do you see. Let's do a very simple thing. Okay? And again, this is not to put you on spot. This is just to get you guys to understand that this is, it's simple. It's okay. It's the simplest thing you can think of. You see a DNA on top? Let me help you. Right? You do. What do you see below that? Histone octamer. Okay. Good job.
As per I am getting it,
Huh.
like ATP is being used and
Where is ATP? जाके दिखाओ. Where is ATP? अच्छा C में नहीं जा रहे अभी हम A में हैं. Only A. What is A?
DNA,
Yes.
then we got the octamer over there.
Uh-huh. Then DNA is wrapped around the octamer. That's what it shows, right? Huh? DNA is wrapped around octamer. They show you a Draven site. You get that or no?
Like broke into part. There is like the second part, B part, it has been broken into parts.
Don't about B. Don't worry about B. Just left side of A. Don't have to worry about the right side of it. You got that one? Good. So what happens in C?
Maybe wrong.
It's okay. There's nothing wrong. It's fine.
I'm getting it like it has been used. That's why.
Okay, what is the first lane? What is the left lane on the blot? Point to the left lane on the blot. Yes, so the left lane on the blot is? Point with your finger. Where do you see the bands? What is that band? The band has a size, right? On the next lane, there is another band.
Size के ऊपर depend डिपेंड करता है
हाँ।
वो लोग कितना आगे बढ़
हाँ,
गए।
कितना साइज है उनका डिपेंड्स ऑन, ओके साइज ऊपर है या नीचे उससे क्या होता है, उसका मतलब क्या है?
रन कर रहे हैं
रन कर रहे हैं तो एक स्लोअर जा रहा है एक फास्टर जा रहा है, कौन सा स्लोअर है कौन सा फास्टर है?
Faster is,
जो नीचे है। हाँ, फास्टर का मतलब क्या होता है? उसका साइज कम है, जो जिसका साइज कम होगा वो फास्टर दौड़ेगा, और जिसका साइज ज्यादा है वो धीरे दौड़ेगा, राइट? एक पतला आदमी जल्दी दौड़ेगा, एक मोटा आदमी धीरे दौड़ेगा।
ये भी सर
Okay, yeah, that's fine. That is,
maybe wrong.
that's correct. No, you're not wrong. That's fine. Let's give you some confidence. It's okay. You guys need to learn to interpret experiments.
Like मैं जितना सोच रही
हाँ,
हूँ they
हाँ,
are, उनका small so they are running faster as compared to
हाँ।
them.
साइज स्मॉल है का क्या मतलब हुआ? ऊपर एक ड्रावन साइट बनाया उन्होंने, न्यूक्लियोसोम में ड्रावन साइट है, राइट? तो अगर ड्रावन साइट कब कटेगा, एंजाइम कब काटेगा उसको? अगर वो एक्सेसिबल होगा तभी तो काटेगा। अगर डीएनए से बाउंड है, did you guys get this right? A simple thing, if DNA is bound to the nucleosome, huh? No, you go there. I want you to be there. It's okay. Huh?
Yes sir.
Do you guys get this right? Simple concept. Because I'm assuming a lot of things. I am assume that you guys get this, and I'm like, oh, when I put you guys on the spot, I think guys that oh, there's like things you're not getting. There's a nucleosome. There is a DNA. If DRA1 site is wrapped to the nucleosome, it's not going to be accessible to DRA1 enzyme, so it won't cut.
Then only it will run.
When it's cutting it, it means it's accessible, it's open, right? So this has unbound, meaning it's right not next to the nucleosome, right? That's the fundamental thing. Okay? तो इसका मतलब क्या हुआ? Yes तो बोल देते तुम लोग, उसका मतलब क्या हुआ लेकिन? हाँ? तो एक फर्स्ट लाइन में क्या है? जस्ट लुक ऑब्जर्व, क्या है उसमें? फर्स्ट लाइन में क्या है?
So they are in the concept.
से पैनल सी में फर्स्ट लेन में लेफ्ट लेन में क्या है? हम लेफ्ट से राइट जाते हैं ना, हाँ, लेफ्ट से चालू कर रहे हैं, राइट?
फर्स्ट लाइन में ये है कि वो एक्सेसिव नहीं है, वो ब्रेकडाउन नहीं हो रहा
ब्रेक डाउन नहीं हो, वेरी गुड। फर्स्ट लाइन में, बाद वाले लाइन में हो गया है, ओके।
है। Then again it is not.
हाँ, तो फर्स्ट टू लाइंस को छोड़ के अब थर्ड लाइन से चालू करते हैं। थर्ड लाइन में ड्रा1 से कटिंग हो रही, कोशिश हो रही है लेकिन नहीं हुआ।
नहीं हो पा रहा है, that's why वो एक्सेसिव नहीं है।
तो फर्स्ट टू लाइंस के ऊपर क्या लिखा हुआ है? इंटैक्ट डीएनए, ठीक है? उसका मतलब उसमें न्यूक्लियोसोम नहीं है, वो खाली डीएनए है, ठीक है? डीएनए को आप काटोगे तो कट जाएगा, नहीं काटोगे नहीं काटेगा, न्यूक्लियोसोम की बात ही नहीं हो रही है, ठीक है? तो कट जाएगा तो वो नीचे दौड़ेगा, नहीं कटेगा तो ऊपर दौड़ेगा।
फिर उसके वो कट हो रहा है, अब वो आगे रन कर रहे हैं।
हाँ, तो फर्स्ट थर्ड लाइन में क्या हो रहा है? कट हो रहा है कि नहीं हो रहा है?
maybe they are trying but
नहीं हो रहा है कट,
नहीं हो पा रहा है।
वेरी गुड। थर्ड लाइन में कट नहीं हो रहा है तो हमने ड्रावन से काटा लेकिन कट नहीं हो रहा है। फोर्थ लाइन में क्या हो रहा है?
They tried and most probably वो कट हो रहा है, but not exactly,
फोर्थ लाइन में कट नहीं हो रहा है,
not exactly, but सर ये
लेन है वो, जिसमें नीचे सिग्नल आपको दिख रहा है वो फिफ्थ लेन है।
सर वो कट नहीं हो रहा है।
तो फोर्थ लेन में कट नहीं हो रहा है क्योंकि क्या है और क्या नहीं है, प्लस क्या है नेगेटिव क्या है? फोर्थ लेन में ड्रा1 है और रिस्क है लेकिन एटीपी नहीं है। ओके, उसके ऊपर पीछे वाला हो रहा है, ऊपर ऊपर